Okay, we'll look at this worksheet now. Now for question 1, a plant is wilted. Okay, so when you see this word, it basically means that the plant has lost more water. Okay, the plant lost more water than what it absorbed. Okay, so when a plant is wilted, the cells, when a plant loses more water or a plant lost more water than what it absorbed, most likely the cells, the plant cells will be plasmolized. Okay, plant cells will be plasmolized. Okay, that's why the plant will then become wilted. Okay, so in this question, they are asking you to identify the likely state of the cells in this plant. So for C, C is a plant cell. If you look at the option B, C, D, all of them are plant cell. Okay, why? Because of the presence of cell wall. Okay, so you can see very clearly. Okay, the cell wall. There is a cell wall in BCD, in the cell in BCD. Okay, all these are the cell wall. Then A is an animal cell because you cannot, there is no cell wall. Okay, there's the cell wall is absent. Okay, so using the cell wall to identify whether it's an animal or plant cell, will be the first step that you can do. Okay, to identify whether a cell is animal or plant cell. So by seeing whether the cell wall is present or not, you can tell. Okay, then if we look at the options, A is actually a shrunken animal cell. Okay, we can also use the word cremated. Okay, but it's animal cell. So a plant don't have animal cell. So we know A is out of the picture. Okay, A is definitely not the answer. So for this question, we are looking for plasmolized plant cell. Okay, so the answer is C. Because in C, you can see that the cell membrane, in this case, this is the cell membrane. The cell membrane is detached from the cell wall. Okay, cell membrane detached from cell wall. So this is characteristic of a plasmolized plant cell. Okay, so I know C is a plasmolized plant cell. So obviously the answer will be C. Then for D, you see that the plant cell is actually a bit rounded, a bit larger in size. Okay, so that will be a turgid plant cell. Okay, so again, turgid, we use it to describe plant cells. Okay, then a B will be a normal plant cell. Okay, not turgid, not plasmolized. So we will just call it a normal, a plant cell in a normal state. Okay, maybe I should just write plant cell in a normal state. Okay, plant cell in normal state. Okay, because it is not plasmolized, it is also not turgid. Okay, so that will be plant cell in a normal state. Okay, so in this, for question 1, the answer is C. Now let's look at question 2 now. Okay, which row shows the correct description for the movement of molecules in diffusion and osmosis? Okay, we know that this question is actually testing you on whether you can remember the definition of diffusion and osmosis. Okay, so a recap, diffusion is the net movement of particles, from a region, from a region of higher water potential, from a region of higher concentration, net movement of particles from, so it's a net movement of particles from a region of higher concentration to a region of lower concentration. Okay, so this is the definition for diffusion. Diffusion is the net movement. Okay, is the net movement of particles from a region of higher concentration to a region of lower concentration. So I can actually replace this from a region of higher concentration to a region of lower concentration with another phrase. Okay, I can actually say down a concentration gradient. Okay, down a concentration gradient. So this is the definition for diffusion. Then for osmosis, okay, osmosis is the net movement. So for osmosis, we are interested in water molecules only. We are looking at water molecules only. So osmosis is the net movement of water molecules. Okay, it's the net movement of water molecules from a region of higher water potential to a region of lower water potential across a partially permeable membrane. Okay, diffusion has to be across a partially permeable membrane. Okay, to a region of lower water potential across a partially permeable membrane. So the membrane must be partially permeable. If it's a fully permeable membrane, then it is not osmosis. Okay, so it must be a partially permeable membrane. And likewise, from a region of higher water potential to a region of lower water potential, this we can replace with down a water potential gradient. Okay, so this one is down a water potential gradient. And yeah, so in this case, the best answer will be A. Okay, because for the other options, it's wrong. So it must be down water potential gradient. So if you do the elimination, A is the possible answer, is the only possible answer. Okay, let's look at question 3 now. Okay, for question 3, let me minimize this first. Okay, now an experiment was set up as shown. The liquid level in the funnel was labeled X. So the liquid level is here. Now the liquid level in the funnel was noted to increase after some time. So it increased. So after some time, the water level increased. Okay, liquid level, not water level. Okay, the liquid level increased. So when the liquid level increases, what actually caused this liquid level to increase? Okay, the liquid level increase is because there is the volume of water in funnel increase. And the volume of water in funnel will increase if there is a net movement. Okay, if there is net movement. Okay, if there is net movement. Okay, net movement of water molecules into the funnel. Okay, from where? From the beaker. Okay, so net movement of water molecules into funnel. And this can only happen if the liquid inside, okay, the liquid inside the funnel, liquid in funnel has a lower water potential. Has a lower water potential than the liquid in the beaker. Than the liquid in the beaker. Okay, then there will be a net movement of water molecules into the funnel. Of course, by osmosis. Okay, the process will be by osmosis. Okay, so let's look at the options given. Okay, which one fulfill this criteria, whereby the liquid in the funnel has a lower water potential, whereas the liquid in the beaker has a higher water potential. So liquid in the beaker, okay, will have a higher water potential. Okay, so which row shows the possible contents in the funnel before the experiment started? So you need to choose the pair whereby in the beaker is higher water potential. So we know in beaker must be higher water potential. Then in the funnel, it must be lower water potential. Okay, so if we compare 10% and 10%, they have equal. Both have the same concentration of sucrose, 10%. So both equal water potential. So I know A is out of the picture. Okay, then if you compare distilled water and 5% sucrose, distilled water have a higher water potential. 5% will be lower. So you know this is B is also out of the picture. Okay, then if you compare 5% sucrose and 10% sucrose, 5% sucrose is higher water potential than 10% is lower water potential. So again, C is out of the picture. Okay, so D is the answer, because distilled water is actually purified water that has been purified using distillation. So by right, it should have no dissolved substances. Okay, no dissolved substances or minimal dissolved substances in it. So distilled water should have higher, okay, will definitely have higher water potential compared to 5% sucrose. Okay, so that's for question 3. Now, question 4 is similar to question 1. We are looking at wilting. Okay, so this is only possible when the plant cells is plasmolysed. Okay, this one I have explained already, so I'm not going to explain it again. Maybe I'll just touch on shrunken. Whenever we use the word shrunken, we use it to describe animal cell. Okay, in your syllabus, we use shrunken to describe animal cell. Shrunken, or you can say crenated. Okay, another word you can use is crenated. Okay, it has similar meaning. Okay, then when we say burst, usually again we are describing an animal cell. Okay, because our animal cell does not have a cell wall. Animal cell does not have a cell wall. So when too much water enter, the animal cell by osmosis, eventually the cell membrane will not be able to hold the contents of the cell anymore, and it will burst. Okay, so usually when we say we use burst to describe an animal cell. Okay, if it is placed in a solution of higher water potential. Okay, so that's for question 4. Question 5. Which structure in a cell restrict the movement of substances into and out of the cell? Okay, this is obviously describing the cell surface membrane. Okay, now the cell wall, the function of the cell wall is to prevent the plant cell from bursting. Okay, you can say prevent plant cell from bursting. Prevent plant cells from bursting. Yeah, so usually you can say this. Then cytoplasm is where, okay, is the place or is the site where most chemical reaction occur in the cell. Okay, most chemical reaction occur. Then nucleus, the function of the nucleus, it controls all activities in a cell. It controls all activity in a cell because it contains the DNA. Okay, it controls all activities in a cell. Okay, why? How come it can control? It's because it contains DNA. Okay, it contains DNA. Can And for question 6, which role explains the difference in results when plant cells and red blood cells are placed in distilled water? Okay, so when you place cells in distilled water, okay, the distilled water have higher water potential. Definitely, it will have higher water potential than the cells. Okay, so the cells will have lower water potential. So there will be a net movement of water molecules into the cells. Okay. However, okay, the animal cell will burst. Okay. By the way, red blood cell is an animal cell. Red blood cell, animal cell. Okay. Plant cell is a plant cell, obviously. Okay. So animal cell don't have a cell wall. So when there is a net movement of water molecules into the red blood cell, eventually the amount of the volume of the cytoplasm will increase to a certain extent whereby the cell membrane cannot hold the contents anymore. And that is when the cell, the animal cell will burst. However, in the plant cell, okay, plant cells have cell wall. Cell wall is rigid. Okay, remember, cell wall is made up of cellulose. So cellulose is a very strong and tough, strong material. Okay, so cellulose is a very strong material. So it will prevent the cell wall, it will prevent the plant cells from bursting. Okay, so the difference in result will be plant cell will not burst, plant cell will become turgid, whereas red blood cells will burst. Okay, because red blood cells is an animal cell and red blood cells, being an animal cell, will not have cell wall. So in this case, it's the cell wall, okay, that cause this difference in the results when you place plant cell and animal cell into distilled water. Okay, and question 7. A plant cell is placed in a concentrated salt solution. The cell membrane pulls away from the cellulose cell wall. So obviously you have a plasmolysed plant cell here. Okay, a plasmolysed plant cell. Now, for this question, you need to remember, what you need to remember and recall is cell wall is fully permeable. So everything, all substances can pass through it because it's fully permeable. So if this cell is placed in a concentrated salt solution, okay, it will be able to pass through the cell wall. So all this empty space will be concentrated salt solution. Okay, will be concentrated salt solution. So the only possible answer will be B. Okay, then another thing that you need to remember that may be helpful for this question is cell membrane is partially permeable. Cell membrane is the one that will control, okay, what particles can enter the cell, what particles cannot enter the cell, because cell membrane is partially permeable. Okay, and that's for question 7. Now for question 8, you have three different Petri dishes, X, Y, and Z. Then each of them is either filled with distilled water 0.1 mole or 1 mole. Okay, so 1 mole per dm cube. Okay, mole per dm cube is a unit for concentration. Okay, it's an unit of concentration. So the higher the value, the more concentrated the solution is. Okay, so in this case, 1 mole per dm cube is more concentrated, or I can say it's a higher concentration than 0.1. Okay, 0.1 mole per dm cube is less concentrated or diluted. Okay, less concentrated, diluted, or I can say is lower concentration. So if you place them into three different solutions, okay, if you place them into three different solutions, of course the length, there will be changes in length. Okay, when will it increase? When will it decrease? Okay, so when the length increases, okay, length increases is when there is net movement. Okay, when there is net movement of water molecules, net movement of water molecules into the cells. In this case, it's potato cells, into the potato cells. Now, if the length decreases, that means there is net movement of water molecules out of the cell. Net movement of water molecules out of the potato cells. So in this case, they use the word respectively. Okay, so Z will most likely decrease. Okay, Z will decrease in length. Then the one that is submerged into or immersed into distilled water should increase. So in this case, the best answer is B. Okay, and let's look at question 9. Cells responsible for absorption of substances are modified to increase their surface area to volume ratio. Which rule does not describe such modification? Long, narrow protrusion of red hair cell is to increase surface area to volume ratio. Okay, the microvilli in the epithelial cells in the small intestine is to increase the surface area to volume ratio. Okay, biconcave shape of red blood cells is also. Okay, long and coiled tube. This one not increase the surface area to volume ratio. Okay, this one actually is to increase the duration. Okay, increase the duration. Increase duration of food in, okay, increase duration of food in the tube, in the esophagus. Okay, so with that, I think I can end this recording.